My name is Camilla Dominighetti. I am the Biology Manager for Cell Line Development at Advanced Instruments, and this is my colleague.
I'm Rick Zampa, and I am the Field Application Specialist for the biotech osmometer portion of our portfolio. So, Camilla?
So for my part of the talk, today I will be addressing the issue of genomic stability in iPSCs, the new approach for restoring cell lines. So here is a brief agenda for my part of the talk today. So I'll start off by giving you an overview of iPSCs and their applications and market research. I'll then discuss some of the challenges associated with handling iPSCs. And then finally, I will discuss some novel solutions. So I'm going to start off with some of the basics. So I'm sure most of you will already be aware of a lot of this information. So the induced pluripotent stem cell technology was first established in 2006. And this was based off the hypothesis that the genes which are important for embryonic stem cell function could also induce an embryonic state in adult cells. So I'm sure most of you are aware of what induced pluripotent stem cells are. They're also known as iPSCs, and these are a type of pluripotent stem cell that can be generated directly from any somatic cell. So this could be things such as skin fibroblasts or blood. And this takes place using a reprogramming process. So this reprogramming process means that we can create a cell population that is capable of indefinite propagation. And we create an unlimited cell supply that has the ability to be converted into any cell type in the body. So what are the key iPSC applications and research areas? So human iPSCs are now being used across a range of different market areas, and this includes disease modeling, drug discovery, and also cell therapy development. Since the advent of the iPSC technology almost 20 years ago, there's been an enormous increase in the use cases for iPSCs, particularly in relation to stem cell biology, but also for regenerative medicine. Alongside this, there's also been enhancements in relation to the cell culture conditions that are being used when you handle iPSCs, and this has increased over the years as well. The combination of these two things has meant that there's been more papers are now citing iPSCs, and they're being more regularly used. One kind of key factor that underpins all of these kind of areas that I've mentioned on the screen is the need for using high-quality cells, and with that, the need for a good QC and characterization process. So let's have a look at some of the challenges associated with handling iPSCs. So as I mentioned earlier, there have been some significant improvements in the culturing techniques for when you handle iPSCs. And this comes down to things such as weekend-free feeding medias, the development of iPSC reagents and also matrices, but they still remain very challenging to culture, and they are also very expensive, especially when you compare them to other cell lines. So one kind of key area that's important when you're handling iPSCs is to have a well-established protocol. And this is because they are prone to different experimental conditions, and they also have issues in relation to reproducibility. So with this in mind, it's becoming more evident that we need a well-established QC protocol. And the good news is that there is a lot more awareness now in relation to the need for a good QC process. And one kind of key contributor to that is the International Society for Stem Cell Research. And they have produced a range of different publications that support scientists when they are handling iPSCs. So in recent years, last year, they published the standards for human stem cell use in research. And this particular document outlines some key areas as to why QC is so important in relation to iPSCs and provides some good guidance for scientists if they are planning to do so. So when we talk about iPSC and QC and characterization, what do we mean? So there's lots of different tests and assays available on the market, but you can kind of categorize them into three key areas. The first is the kind of general QC, and this relates to things such as cell hygiene, so to ensure that there's no fungal or bacterial contamination. It also relates to the growth of the cells to ensure that they are growing at the normal rate as you would expect, and they are reaching the normal doubling times. And finally, the morphology. So you need to make sure that your cells are growing and looking as you would expect them to. The second category is the functional assays. So this is to ensure that your iPSCs are behaving as they should and as you would expect them to. And it needs to make sure that you are checking that your cells are expressing the correct markers. And then finally, we have the genomic stability. And this is something that I'm going to discuss a little bit further. So as I mentioned earlier, there's lots of different assays available on the market, and this also relates to genomic stability. So it can be quite difficult to understand what assay to do and when. And the genomic stability assays look at a range of different things. This includes copy number changes, genomic alterations, and single nucleotide variants. So on the screen here, I've kind of given some key factors that you should consider when you are looking to run genomic stability assessments on your iPSC cells. So this includes things such as your budget, your time frame or your urgency, and what is the intended use of the cells. It also includes where you are in your project and your project timeline. Have the cells gone under any specific stress, such as cryopreservation? And have you noticed any change in their behavior? One key consideration when you're looking at genomic stability is the detection of mosaicism. And this is where we have a single nucleotide variant that's not present in all of the cells. And the risk here is that as time passes and you culture your cells with known mosaicism, there is a risk that these cells could have a growth advantage over the wild type. So it's estimated that between 20 to 30% of all iPSC cell lines have an unwanted change or mutation. And some of these are in the common 20q11.21 copy number variants. But as iPSCs are more regularly being used, we're now seeing this list of mutations increase. And there are a lot of risks associated with these additional mutations. And this can include things such as changes in the cell behavior, the ability for the cells to grow, and the ability of the cells to differentiate. And although we have to continue making this list and ensuring that they are being identified, it's making it quite clear that there is a need for a good QC process. So I'm now going to discuss some novel solutions, and this data has been generated by a company called Selected. So as I mentioned earlier, we know that 20 to 30% of iPSC cell lines are likely to have an unwanted change. And historically, scientists only have a couple of options when this takes place. So routinely, most people will consign these cells to the clinical waste. And this therefore means that they need to start their project again. And in many cases, they have to get a new cell line or refer back to a previous bank or passage. In general, these are very risky and time intensive. It can lead to very long lead times in relation to your projects and add additional cost onto your project. So with this in mind, Selected researched a few different options in relation to how you can rescue a cell line if you know that there is a genomic abnormality. And the key area of research that they have investigated is the use of single-cell cloning. So traditionally, single-cell cloning is often used to identify perhaps a gene-edited or mutant clone within a primarily wild-type population. But in this instance, they decided to flip this over and look at trying to find a wild-type clone in a population which was predominantly mutant. So over the last year or so, Selected has been optimizing and validating and developing their rescue platform. And this employs the Advanced Instruments VIPS and Cellmetric X instruments, and it enables them to rescue cell lines even when the wild type is minimal within the population. So during this process, they single-cell seed using the VIPS automated seeder, and this ensures that they get a high number of clones. They then track the growth of these clones using whole well imaging, and they can do this across a specific time point, and it's documented. They then select the top clones that they desire based off the clonality which has been confirmed, and they then run QC to ensure the quality of the cells and to ensure that they have been successfully recovered. So as part of this process, they ran a few different rounds of protocol optimization. So they split this into 5 key areas. The first is they had to ensure that the single-cell seeder could correctly identify the iPSC cells. From here, they then needed to ensure that they were able to monitor the growth through whole well imaging and ensure the cell detection at that point. They then looked at the clonal outgrowth and the success of this, followed by the media and reagent optimization. And finally, from all of the above points, they were able to develop a robust cell handling process. So you can see the data on the screen here represents the different conditions and optimization processes that they completed. In addition to this, they also spent quite a long time optimizing 384 well plates in relation to iPSCs. So historically, seeding and growing iPSCs in 384 well plates has been quite challenging. And this is because of the low media volumes and the risk of evaporation, but also because trying to coat 384 well plates with the matrices can be quite difficult. As part of this process, they tested a range of different reagents and compared the 384 well plate clonal outgrowth against the control, which in this instance was the 96 well plate. And what you can see here on the graph is that reagent 2, in combination with the 384 well plate, provided the highest clonal outgrowth, which was very close to 80%. So from these clones, they needed to ensure that the quality of them was desirable. And it was therefore critical that they ran the immunocytochemistry testing to ensure that they were still expressing the correct markers. So from this data, they ran multiple different plates across multiple different cell lines and reagents and looked at fixing the plates and running the different markers with the DAPI control. The data from all of those show that they had no loss of expression and they were able to continue on with their research. So I'm now going to discuss 2 case studies using the rescue platform which were generated by Selected. So the first case study looked at a pool of cells that had a known low mosaicism between 20 to 30%. So this meant that they knew that 20 to 30% of that pool had clones which had a mutation. From here, they seeded using the single-cell seeder to achieve a high number of clones. And after 2 hours, they imaged on the cell metric X for single-cell detection and confirmation of clonality. The image you can see on the left-hand side shows at day 1 that the cell has divided into 2 cells. And the image on the right-hand side shows the colony which formed from those cells after 7 days of incubation. So selected during this period, they scanned the plates routinely to ensure that they could monitor the growth and ensure that the morphology of the cells was as they expected. What the data from here showed is that they were able to select 10 top clones which had confirmed clonality, that they could move on to the later stages of expansion and for further QC and eventually cryopreservation. So as part of this, they had, during the expansion process, they looked at 8 of those clones, and using digital PCR, they were able to extract the DNA and look at whether they were wild type or if there was a mutation present. 5 of those clones were wild type on the 20q11 locus, and 3 of those clones showed a mutation at that locus. All of the clones which were confirmed wild type and confirmed for clonality were monitored for morphology, and the cells were expanded, and they were able to preserve the cells, and the cell line was rescued. The second stage of the case study was looking at a predominantly mutant pool. So this particular pool had a no mosaicism of around 90%. So this meant that in the early stages of their research, they had to single-cell seed a lot of clones to ensure that they had a high number at the downstream process. So they followed a very similar outgrowth to the previous case study, and with this they looked at a range of different plates, and you can see on the left-hand side image that they achieved around 50% clonal outgrowth from a representative plate. Again, they confirmed clonality of those cells. They monitored for clonal outgrowth. They tracked after incubation. And then they were able to select 20 of those clones to move forward to expansion and QC. And for this particular set of clones, they ran a SNP array analysis. And they were able to identify that 19 of the 20 clones were mutant, and only 1 of those clones was a wild type. So again, they followed a similar procedure. They were able to monitor the morphology to ensure that the cells were growing as they expected and that they morphologically looked as they expected. From here, they expanded the cells, they confirmed the cryopreservation, and this cell line was rescued. So here I've outlined some of the kind of key advantages of this rescue workflow. So the first is that it enables optimized protocols. So Selected have spent a very long time optimizing the protocols, especially using automation, and they can now achieve very high clonal outgrowth. This means that they have a decreased number of required plates, definitely in the early stages, and this evidently means you have fewer consumables and less laboratory time. The addition of automation meant they could have a higher number of single cells, and again, this led to a fewer number of plates required and a reduced time spent in the laboratory. And finally, the ability for them to run the in-process QC meant that they could run rapid analysis and triage earlier. This meant only the optimal clones were selected and cryopreserved. So to summarize, iPSCs are now being used across a wide different range of market areas, and they're known to be critical for many workflows. They still remain very challenging to culture and to grow. However, there are some improvements that are being made. The issue with genomic stability is well known, and people are now starting to release documents to support scientists in relation to that. And the final point I want to make is that it's known that genomic stability is a growing problem, and we know that up to 30% of our current iPSC cell lines could have a mutation, and therefore QC and characterization is key at this point.
I have a question for Camilla about the outgrowth that you showed. At 96 well plate compared with 384 well plate, do you see a difference?
So from the data that Selected generated, there was an increase in outgrowth for the 384 well plates, and this was with the certain reagent 2. So it would be dependent on the conditions for the outgrowth, but in that particular case, the 384 well plate had a higher outgrowth.
That's what I observed. So
Yeah.
I wanted to see the component that you used was different in 96 well plate versus 384, right?
Yes,
Okay.
yes.
Thank you.